All right, the cell cycle's got to be pretty tightly controlled, because if it's not controlled, what do you get? The opposite of controlling the cell cycle. Cancer. Okay, cancer is the cell cycle out of control. So we need to closely control it. One of the ways that it's controlled is through what are called checkpoints. A checkpoint is just what it says it is. The cell is checking to make sure that everything's right. And if it is, it says go to step 2. If everything in step 2 is right, go to step 3. But there's different ways to check and stop if something's not going right. The first checkpoint is near the end of G1. And at the end of G1, checks to make sure everything's ready to go. If it is, this is called the point of no return. Once a cell goes past G1, it's either going to divide or what? Die. One of the two. Okay? There's no going back. There's no going into G0. It's either going to divide or die if it passes this first checkpoint. We know that during the S phase of interphase, that's where the DNA gets caught. After S phase, we go into G2. That's where our second checkpoint's at. It's near the end of G2. G2 is the G2 checkpoint looks to make sure that number 1, there's 2 complete sets of DNA, and number 2, there's 2 complete sets of organelles. If you've got 2 sets of DNA, 2 sets of organelles, the G2 checkpoint is passed, and you go on into mitosis. The last checkpoint is actually at at metaphase. Sometimes it's called the M checkpoint, but it's at metaphase. And what signals the checkpoint to proceed from here is if all the chromosomes attach up to the spindle fibers. Once they're all attached, all 46 chromosomes in a human are attached to spindle fibers, that tells the M phase that, okay, this checkpoint is passed, finish this up. Go ahead and divide. So the 3 checkpoints are at the end of G1, at the end of G2, and at the metaphase stage of mitosis. Once all the chromosomes are attached to spindles, that means go ahead, finish mitosis, do cytokinesis, split this cell. Now we're going to go and get into a little bit of stuff that's sort of complicated here in the next part. And we're going to watch a video in a minute, another little clip about how this works. But you have 2 main things that help to tell your cell to go ahead and divide. And those are 2 chemicals, one called cyclin, and another one called CDK. CDK. That chemical, CDK, stands for cyclin-dependent kinase. And we talked about kinases over and over again. Kinases are enzymes that do what? Phosphorylate things. They add phosphates. And when they add phosphates, what else do they typically add? Energy. And what does that usually happen to the thing that got phosphorylated? It activates it. It makes something happen. So these CDKs actually turn on mitosis by phosphorylations. I'm going to stop this here for a second, and we're going to watch that video clip, and then I'll come back and sort of fill in the gaps after the video clip. Some did, some didn't. All right, so let me go back over kind of what he was talking about. He mentioned 3 things. He talked about cyclins. Cyclins are chemicals made during the cell cycle. They help to control the cell cycle. Cyclin. Yeah, it is. He also talked about CDK, which was cyclin-dependent kinase. These 2 things have to get together. Cyclins and cyclin-dependent kinase. The further in the cell cycle you get, what happens to the concentration of cyclins in the cell? It's higher, which means it's easier for it to find what? CDK. When they get together they're called MPF, maturation promoting factor. This MPF is an active kinase. It goes out and phosphorylates the nuclear lamina, which is the part of the nuclear envelope that holds it together. When it gets phosphorylated, what happens? It falls apart. And that's the start of what? The nuclear envelope falling apart is the start of what? Mitosis, prophase, prometaphase. That's where it starts. So That's important. It's important that you understand that. And one other thing I want to mention is when cells stop growing. Most cells, normal cells, have what's called density-dependent inhibition. What that means is when they bump into each other, what happens? They stop growing. That tells them, hey, stop. When one cell bumps into the next, it means stop. That's the way normal cells work. Now, do cancer cells do that? No. Cancer cells bump into each other, they just start stacking up. And we call that big stack of cells what? Tumor. Also, most cells have to be anchored to something underneath to grow. Because underneath is where they're going to be getting what? Their nutrient, their blood supply. It's going to be coming from underneath them. So if they're not attached, they die. They're anchorage dependent. Now with cancer, we know cancer is all about abnormal cell growth. Turns out that cancers have a lot of cyclins, and cyclin activity is high in cancer cells. And also that enzyme telomerase is working in cancer cells. Remember that most cells are programmed to replicate a certain number of times and die. What's the word for that? Program? It's a word you need to know. It's called apoptosis. And they know it's time to die based on the length of what? Telomeres, which are those extra pieces of nucleotides at the end of every chromosome that get shorter every time it replicates. Normally, the only time this enzyme right here, telomerase, would be active is when? In you, when's the only time you want telomerase working? When you're an embryo. It makes your chromosomes get longer so you won't lose what during replication? Genes. Okay? It's extra pieces. But in cancer, one of the mutations that causes cancer is that this gets turned back on. And since this is turned on, that means your cells never what? They never die. The apoptosis never happens. So cancer cells basically become immortal. As long as they have oxygen and nutrients, they keep dividing, and they will forever. And that's a problem. That means cancer's not going to go away on its own, because those cells aren't just going to die off. They're going to keep going. So you have to have a way to do it. I want to mention two other words, and then I'm going to stop for today. We know that cancer can be caused by a lot of things. It can be caused by chemicals. It can be caused by genetics. It can be caused by radiation. Some forms of cancer can be caused by viruses. And the cancer comes in two kinds: benign cancer, which means you get a tumor, but it doesn't what? It doesn't spread. It stays in that spot. And basically, you can just cut it out, and you're going to be okay. You're going to get surgery, and you're probably going to be okay. The other kind of cancer is malignant, and that means it does spread. And a word I want you to make sure you have on your notes, and you know the definition of, and we'll end with this word, metastasis. Or sometimes it's written as a verb, metastasize. Malignant cancer spreads. That's what metastasis means. It spreads. It's cancer that spreads from one part of your body to another. What two ways does it usually use to spread? What two highways through your body? Your blood vessels. And what's the other system of vessels in your body? The lymph vessels. That's how it metastasizes. It travels through those. All right, I'm going to stop that there.